Thanks for joining. Casey's managing the in-person participants today. Thank you, Casey. Okay, so this morning we are very excited to have Allison Post speaking to us about some of her work with PhenoCam. For anybody who doesn't know Allison, she's the program manager for Earth Lab. She has a bachelor's degree in biology from the University of Maryland and a PhD in ecology from CSU, Colorado State University. And she recently did a postdoc at NAU, Northern Arizona University. She's an ecosystem ecologist by training, who studies how climate change impacts terrestrial ecosystems, including grasslands. She's worked before with large-scale field experiments, looking at how grasslands respond to changes in precipitation patterns. And she is going to tell us today a little bit her work with PhenoCam to look at how climate influences the timing of seasonal transitions across diverse grassland ecosystems. Okay, thank you, Allison. You can go ahead and take it away.
Great, thank you, Chelsea, for that introduction. Okay, I'm gonna figure out the dance of sharing the correct screen here. 1 second. Okay, can you guys see that? Is that a yes?
Yep,
okay.
Looks good.
Great, thank you. All right. Yeah, as Chelsea said, I'm Allison. I am the program manager for Earth Lab, but I just completed a postdoc at Northern Arizona University with Andrew Richardson, who is the founder of the PhenoCam network. And it's been really fun to kind of go from field work to this more continental scale digital photography data sets. So I'm excited to first explain what the network is and then show you some of my research with it. All right, so here's the overview for what I plan to do today. I'm going to do an introduction of the PhenoCam network, what it is, how it works, how it's established. And then I'm going to actually just show you how to navigate the website, because it's a really cool feature that you can see all this data in real time on the PhenoCam website. And then last, I'm going to give just an example of a study that I've done using this PhenoCam data. So let's just start with the definition of phenology, make sure we're all on the same page. So phenology is the scientific study of periodic biological phenomena, such as flowering, breeding, and migration, in relation to climatic conditions. So it can be what I tend to think of as in changes in vegetation. So when leaves bud out in the spring, or flowers bloom, or when they senesce in the fall, they change colors in the fall. But it can also apply to animals, such as when animals migrate, when insects do metamorphoses, such as caterpillars into butterflies. So it's anything in biology that happens cyclically. And it's a really fun thing to watch through the season. So for example, this is the phenology across the Earth from satellite imagery. So you can see the seasons changing. Some people refer to it as the pulse of the Earth, the rhythm of the seasons. And you know, historically and into today as well, people have been fascinated with how seasons change and all the things that come with that. So it's a really fun thing to think about. However, climate change is actually affecting when these seasonal transitions take place. So for example, due to warming temperatures, spring phenology is occurring earlier. So for example, leaves coming out in the spring, and autumn events such as leaf color change are happening later in the season as well. And this is resulting in an overall lengthened growing season with cascading effects on carbon, water, nutrient cycles, anything that is related to these phenological events. And for that reason, the IPCC concluded that phenology is perhaps the simplest process in which to track changes in the ecology of species in response to climate change. And I just wanted to show you guys this. This is actually from the National Phenology Network, which is a citizen reporting system where you can say when things are budding out where you live. And they just released this map just last week showing how early spring leaf out is occurring across the eastern half of the US. You know, in the western half, we've been abnormally cold this season. The eastern half has been abnormally warm. And so I'm going to see if I can get this video to play here. And it's going to show the anomaly in spring bud out. So red means it's really early, and blue means it's happening late. Yeah, it has cool music with it too. But what is really interesting to see, I mean, that means all that red there, those areas are budding out in the spring up to 20 days early. So that's really early because they've had really warm temperatures for the most part. For example, I grew up in the Washington, D.C. area, and they had buds and flowers on the trees in late February, which I've never experienced in my life growing up there. So it's crazy to think about how much the seasons can be altered. So in addition to what I just showed you, there's many other consequences of changes in the patterns of phenology. For example, you can have more extreme weather events, such as deluges or wildfires. You can have increased allergens. It can affect tourism, right? So people go to leaf peep, but if you don't know when that's going to happen, or if it doesn't have as long of a period to change color, then that's a shorter window to see it. It can affect agricultural crops, insects such as mosquitoes, nuisance insects, but also good insects such as bees that might mismatch in timing with their pollinator or the species that they pollinate, for example. So there's a lot of consequences and changes in the timing of these cyclical events or changes in phenology. So in order to track these changes in phenology, our research group established a network of digital cameras to automatically monitor changes in plant phenology. And this is what we call the PhenoCam network. And what we did is we and many collaborators set up commercially available digital cameras that take images every 30 minutes during the daylight hours. And also some of these cameras can also take images in the IR spectrum. And we use this imagery to monitor vegetation phenology. And what it does is it gives us a direct link between what's happening on the ground, so what we see as individuals, and what is seen by satellites at a much coarser resolution. And it's grown. So it was established in 2008, first at a site in Bartlett near Harvard, or Northeast. And we currently have over 700 cameras, a lot of them in North America here, but we're starting to get a wider following as well. So more cameras in Europe and Asia and Australia, South America. And what's really cool is that all this data from these images or from these cameras are available on the website in real time. So if you go to this website, which you can try out later, you can look at any of these sites and see what it currently looks at that location. I wanted to point out for this crowd too, that all of the neon towers have a PhenoCam associated with them. So wherever there's a triangle here, that is a neon tower that had a PhenoCam installed on it. So it's very integrated with neon data. And what's great about that is you can then associate these images with all of the other data that neon collects at those locations. All right, so time to look at some pretty pictures. Here's just a sample of a variety of ecosystems around the world that we have PhenoCams for. You can see there's agricultural land, there's grasslands, shrublands, wetlands, desert, tropical rainforest. It's really fun to look through them, and sites are being continually added as well, which is cool. But also you have a variety of ecosystems, but it's also fun to look at one ecosystem throughout the different seasons. So this is a site near Quebec, Canada, and it's my favorite because of the fall leaf colors there. But this is how it looks during spring, summer, autumn, and winter. It's just a really striking difference. And since we're all in Colorado, or will be soon, I wanted to highlight some Colorado cameras. We have one established at Rocky Mountain National Park. We have a couple established at Niwot Ridge. So this is tundra and then evergreen forest. We have one at Rocky Mountain Biological Laboratory. This is an example of an aspen stand there. There's an agricultural farm out in Sterling, Colorado. And there's grassland at the Central Plains Experimental Range. That's where I did all of my PhD work. And so we have a nice variety of ecosystems represented here in Colorado. I was hoping there'd be a cool boulder camera, but there's really not. This point per boulder looks like it was someone testing a camera, so we only have a few images of a parking lot. But I think that just means there's an opportunity to add some cameras around Boulder. I think that would be really cool to do. So you've probably heard the saying that a picture is worth 1,000 words. And that's exactly true for PhenoCam images. They can tell the story of an ecosystem. So here I'm going to go through some before and after images of some extreme weather events or things that have happened at some of these sites. So for example, this is a beautiful site in Page, Arizona, just north of where I am in Flagstaff. You know, it's a cold winter day. It looks like you might want to go to this warmer location and hang out, but maybe not if there's a dust storm. This is what it looks like during a dust storm. Again, maybe you want to go on a tropical vacation to Puerto Rico. Looks nice there, but maybe not during a hurricane. So this is a before and after of when a hurricane came through this site, and you can see a lot of the leaves just got ripped off the trees. Here's a nice lush grassland in Central California. And this is what it looks like just 1 week later after cows were allowed to graze on it and leave some other evidence of their presence as well. And here's an example of a wildfire in an Oregon evergreen forest. So this is August 19th, 2020. And this is what it looked like on August 20th, 2020. So just a day later, you can see there's still some smoke in the air, and some of these trees immediately died from that wildfire. Here's a controlled burn in a forest in Florida. So look at the understory here. We can see that it turned a dark black color from being burned. But that was all doom and gloom. There's also pictures of recovery. So I wanted to include this one. This is the redwood forest near Big Basin. And if you remember in 2020, there were the CZU complex fires that burned through these redwood forests, which was really sad. But actually our research team went and established some PhenoCams at that site afterwards to look at how recovery happened. So this is what the redwood stand looked like in April 2021. And here I'm happy to report is what it looks like in current day. So you can see there's a lot of growth happening again on these big redwood trees. A lot of what they call epicormic growth. So from the stems and the branches, new shoots are coming out, which is really cool. And just a local example, Flagstaff has had record snowfall this year. So this is an example of a snowstorm that we had where I am located here. And you can just see how things get buried. So I want you to especially notice this little pine tree in the foreground. This is January 14th of this year. January 15th, it's getting snowier, pine trees getting buried. January 16th, the camera is also getting buried. And then finally it stopped snowing on January 18th. We had about 3 feet of snow from that event. So it's fun to see these storms play out in real time as well. All right, okay, so that was a lot of pretty pictures to look at. But PhenoCam imagery is more than just these pretty pictures. We can also extract quantifiable values from them. We can extract a value of greenness from the vegetation. How do you ask? Well, do you ever remember sitting way too close to a TV as a kid? You probably saw pixels if you did that. Everything got really blurry and all pixelated. Maybe something like this. Well, if we zoom in on those pixels even more, you can see that each pixel is made of red, blue, and green. And when they're viewed further away, they combine to create the appearance of any color. And that is because of what's called the red, green, blue additive color model. And this just states that we can reproduce any color through a combination of these three primary colors of light. And for example, you have red and green can make yellow. And I want to point out that light mixes very differently than solid pigments, because if you mixed red and green together, you'd probably get a shade of brown. But when you mix light, you get nice yellow color. So that is how we can use these three primary colors of light to create any color that we see. And that same concept is used for digital cameras. So just like on TVs, old style TVs, these images from digital cameras each have a numerical value indicating its red, green, and blue intensity. And what we can do with this is we can extract that information from these images. So for example, this is the nice site we had in Quebec, Canada. If we want to extract the greenness, this portion of the image, we call this the region of interest, or the ROI. We can divide this image into three different color channels. So it's red, it's green, and it's blue color channels. And then we can calculate a metric of relative greenness compared to the total brightness of the image. So this is the green pixel intensities over the red, green, and blue all added together. So you get relative greenness of this image. And we call this the green chromatic coordinate, or the GCC. And when you do that over time, you get these beautiful patterns of seasonal changes. So this graph, GCC is the y-axis, the x-axis is year, and you can see that every year we get a beautiful green up in the spring. Then the leaves kind of turn a darker green throughout the season. So that's why you see this slow drop off of greenness, but then a sharp drop in greenness when you get that color change in the fall. And then it does it again the next year. So you get these beautiful cyclical patterns of vegetation phenology. There's another thing that we can extract from this data as well. We use these greenness trends to extract the seasonal transition dates. So such as when spring or fall begins each year. So what you can do is you can use the amplitude of this greenness curve here, and we can determine the day of the year when the site has reached either 10, 25, or 50% of its maximum greenness. And we say this is when spring has started. And you can do the same thing for the fall dates as well. So this is the start of fall. And what's cool is that these seasonal greenness patterns, as well as these transition dates extracted from those patterns, are all available for download on the PhenoCam website. They're just automatically calculated. So I just wanted to highlight a couple other examples or advantages of PhenoCam. For example, we can choose to only analyze a certain portion of the image. So for example, this is a shrubland, and we've drawn ROIs, or regions of interest, around just the shrub part of the field of view. And what that helps with is a lot of satellite imagery struggles with background reflection from bare soil, but we can completely eliminate any signal from bare soil this way. Another advantage is that you can isolate certain functional groups or certain species to look at. So for example, in this image, I wanted to look at just the evergreen trees in the background and not the signal from these shrubs in the foreground. And then the last thing is that you run into many fewer issues with cloudy days because the cameras are so close to the ground that you get pretty continuous data no matter what the weather is. All right, so let's look at a few more pictures. Time to travel to some sites. But this time we're going to see how different ecosystems have very different patterns in their greenness signals. All right, so challenge question. We looked at a deciduous forest and we saw beautiful trends in greenness. What do you think an evergreen forest is going to look like? I know it's hard to interact, so I'm just going to let you think about it in your brain. Do you think we're going to see a difference? What if I show you these pictures? This is winter and summer at an evergreen forest in Niwot Ridge. It looks kind of different, right? Well, and here is the pattern of greenness for this site. Evergreen trees, in fact, do go through a cyclical pattern of greenness throughout the seasons. And they don't lose their needles, right? They're evergreen, they stay green, but they become more intensely green during the summer. So during the winter, they tend to break down some of the chlorophyll in their pine needles, and so they just appear less green. They have less chlorophyll in them during the winter. So that's why we get these nice patterns of greenness in these evergreen forests. All right, this is grassland site at the Central Plains Experimental Range, so northeastern Colorado. This is what it looks like. And something you'll notice is that these grassland patterns are much sharper peaks than what we saw with like a deciduous forest or an evergreen forest. And that's mostly because they're really sensitive to water. So they get some water in the spring, they get as green as they can, and then as soon as they run out of water, they start browning down. They don't maintain that greenness. They're much more narrow, sharp peaks, as you can see here. Something I wanted to point out with this site is this is June 15th, 2021, and this is June 15th, 2022. You can see how much water you get really impacts what it looks like each season. And in fact, 2022 here, last summer, didn't even see a big peak in greenness. So it was a really dry season for this grassland out there in Colorado, which is kind of crazy. Another ecosystem type to look at is shrubland. This is in Sevilleta, New Mexico. Here's what it looks like in the winter versus the summer. You can see those shrubs green up really nicely during the summer season. And a pattern that we see a lot with shrublands is this double peak here. And the reason that happens is because these shrublands, they can use the water from snow melt or water that's accumulated over the season in the soil to green up in the spring. But then in these monsoonal systems, the Southwest, Arizona, New Mexico, they have monsoons. And so it's really dry early summer, and then you start getting rain July, August, September. And so we have green up from water stored in the soil from the winter, they brown back down early summer, and then they green up again when those monsoon rains start and kind of have a second wind for their growing season. So you see that happen in a lot of seasons in these shrubland systems. And then I wanted to show you an example of one that doesn't have nice cyclical patterns every year. This is a desert grassland in Moab, and it really just depends on when it gets water, when it greens up. It's not really cyclical. And as an example, here's the greenest it got in May 2019, but the greenest it got in 2020 was here, and it looks much less green than it did the year prior. And then finally, tropical rainforests also can show some cool patterns. This is a tropical rainforest in Puerto Rico, and they don't have traditional seasons like we do, 4 seasons. They have the wet and the dry season. So temperature stays pretty constant, but during the wet season, you get nice lush trees. But then during the dry season, you get this here, where a lot of the trees actually lose their leaves during that dry season as well. And we see that pattern in greenness. All right, so that was fun to kind of go through some sites. I enjoy looking at them. Now that hopefully you think it's an interesting data set, I want to show you how to navigate the PhenoCam website to look at these sites and download some data yourself. So the PhenoCam website is phenocam.nau.edu. So if you just type that in, this will be the home page. And there's 2 ways to view all the sites. You have the gallery view and the map view. So first we're going to take a peek at this gallery view. And if you click that, you get this list of all the available sites in alphabetical order. And you can click on any of them to get more information about them and to see images from those sites. But you can also search this gallery, which I'll show you in just a second. But something I want to point out here is that we've sorted the sites into 3 different types. And it really just tells you how reliable the data is. So type 1 sites are gold standard. They follow our standardized protocol. They use the cameras we recommend, and we're actively in touch with the site personnel who maintain those cameras. Type 2 cameras, usually everything is the same, but they've used a non-standard camera. So we have one that we recommend. They've used a different type of camera, but it's usually still pretty good data. And then type 3s, they deviate a bit from this protocol. And often we're not even in touch with the people who own the data. They've just allowed us to upload it. An example of these type 3 sites are a lot of the cameras from national parks. So they'll have webcams to look at parking conditions or weather conditions. And we're allowed to grab those images and make them available on our site, but we don't really have any say over what camera they use or how often they take images or anything like that. All right, so if you want to see a site, you just click on a picture. If you want to sort the sites, it says click here. You click there and you get these options. You can sort by site name, the site type, like I just explained, what the primary vegetation type is, so evergreen, deciduous forest, grassland, wetland, and then whether it's currently taking images or not. And we also have a lot of cameras associated with the Ameriflux network. So you can also see if there's flux data available at these sites as well. Okay, so that was gallery view. What I really like to do is go to map view. So if you click that, you get something that looks like this. And this is a zoomable map that you can explore. And I want to point out a couple of features. You can click here for the same site filters that I explained in the last slide. You can get a different base map. So I like to change this one a lot because I like to see the state outlines, because otherwise I get lost where I am in the US. And then down here, they're colored by site type. So the greens are the type 1, yellows type 2, reds type 3. And if you click on a dot, you'll get this little bubble pop up. It'll give you kind of an overview of the site, a sample image of what it looks like. And then if you click on it, it'll take you to the site page for that site. And this is what a site page looks like. So I clicked on that bubble. This is what it takes me to. And you can see at the top, there's a lot of basic site information. So location, how many images are available, the time span the images are available for. You can click site metadata to get even more information, which gives you, for example, mean annual temperature and precipitation, some climate variables like that. This is a zoomable map again to see where this site is located. And then if you want to see the raw images, you click here, click to browse all the images. The most recent image is shown here, which is kind of cool. You can see what it looks like, on that day. And then here is where you go to see and download all of that extracted greenness data and the seasonal transition dates. So it's called the ROI page. Remember that region of interest that we're analyzing for greenness from this image. So first, I'm going to show you what it looks like to browse images from this site. So I'm going to click there, and they're sorted by year first. So you can see this is all the months from 2022. And if I click on a month, you get a nice calendar view of all of the sites. So one image from each day. And if you click on a day, you get to see all the half-hourly images from that day. And I just want to point out that to get down to this level, to see the half-hourly images, you do need to make a free account on PhenoCam, and that just prevents people from trolling all the images and downloading them, and helps us track how many people are using them. But it's completely free to just set up an account on the PhenoCam webpage to be able to actually click and download these fine scale images. Okay, if we go back to the site page, the other thing we could do from it was look at that extracted data from the ROI. So I'm going to click there, and we get something like this. And I want to point out a couple of features here. We see a plot of the greenness data here, and this is an interactive graph. And so there's actually sliders here where you can pull them in and out to zoom in on certain regions or certain time periods in this graph. And each point is also clickable. So if there's, you want to see what it looks like on this day when it was maximum greenness, you can click on that and it will take you to that image from that day so you can see what it looked like. If you want the data, you can click here for provisional data. This will download several CSV files, including all this greenness data and the seasonal transition dates. These are the ROI masks, so the region of the image that is being analyzed for greenness. And you might ask, why are there two of them? Well, if the camera ever moves a bit, we have to draw a new mask to try to keep it on the same section of vegetation being analyzed. So you'll see this for a lot of the sites. This ROI here was used 2015 to 2016, and then something happened, the camera got bumped, and we drew a new one that went from 2016 to present. So that's why there's multiple, we call them masks within an ROI. And that's the general overview of the website. I just wanted to highlight a couple other things in case you wanted to dive into this further. You can download the raw images under Data tab here. There's information about how to set up a new site if you want to join the network. And under more info, we have multiple cool things. We have a tutorial about how to use this website, kind of a more in-depth version of what I just did. We've established some R and Python packages to work with PhenoCam Data. So if you want to download a lot of data, there's some nice R packages to do that much quicker than doing it manually. We also have curated data releases. So every several years, we release all of the data in a static form. So, it's all cleaned and it's not going to change at that point. And then we also have been collaborating with some teachers and educators and have some K-12 lesson plans and handouts that we've developed with them to work with this PhenoCam data. So as you can imagine, it's a really fun thing to do with students because you get to click around and fly over the world and see what things look like, which is really fun. So then I just wanted to highlight a couple things that we do with this data as scientists. So for example, one thing we do often is just compare PhenoCam to both ground level observations and satellite observations. So for example, this is from a site, a deciduous forest site, and we have the green is the PhenoCam greenness, and this kind of gray color is MODIS and DBI. You can see they line up pretty well for this site. You can monitor growth cycles of different crops. So it's fun to see how different crops change throughout their growing cycles. You can track climate change experiments. So this is the Spruce Project in Minnesota. So it's big chambers that warm portions of this evergreen forest and also elevate CO2. And we have cameras established in all of these chambers, so we can see how the vegetation is responding to those simulated climate change drivers. Another thing is you can relate patterns of phenology with things such as location and elevation gradient. So this is, for example, mean annual temperature of a site versus its growing season length, which we could extract from PhenoCam data. And you can model seasonal changes, which I'm going to dive into because that was my project for my postdoc. So we're going into part 3 of the talk now. I'm going to give an example of a project that I did using PhenoCam data titled Predicting Spring Greenup Across Diverse North American Grasslands. So as you've probably figured out from this talk, the timing of these seasonal transitions are really important, and scientists are always trying to model or predict when seasonal transitions will occur. And they affect many other important ecological processes too. So it's important to know the timing of these seasonal transition dates. And so for this talk, we're going to focus on spring transition dates. When does spring start? So the oldest and simplest spring phenology model is called the growing degree day model. And that just states that spring greenup occurs when there have been enough cumulative warm days. And we see that this model works pretty well across a lot of the northern, northeastern portions of North America, where there's a lot of deciduous forest trees. So here we see 3 to 9 day difference between observed green up dates and those predicted by this model. But if we get to the Western US, it's a whole different story. We see 21 to 37 day difference between observed and predicted green up dates. And this pattern is the same for even more complex models. It tends to not predict these Western part of the US very well. And why is that? Well, one thing to first note is that the Western US is largely grassland and shrubland. So this map here shows all the diverse types of grasslands that we have out there. You have the Great Plain, short to tall grass prairies, desert grasslands of the Southwest. You have, montane grasslands. You have annual grasslands out in California. So they're very diverse. And so then the question remains, why don't these traditional spring phenology models that predict when spring occurs work for these grassland ecosystems? Well, we hypothesize that it could be because these traditional models are missing a critical factor for grassland spring green up, and that is water availability. So unlike forests, which usually have ample rainfall, grasslands are inherently water limited. That's why they're grasslands. They don't have enough water for trees to grow. And so this map, for example, shows the primary limitation to plant growth across the world. And I want to highlight that that Western US there in that pink kind of reddish color means that it's mostly limited by water. And so what we decided to do is to use data from the PhenoCam network to develop and test multiple new spring phenology models that incorporate a measure of water availability for North American grasslands. So this map shows the locations of all the sites that we used. We had a total of 43 sites and 195 site years. And the size of the circle shows how many data years were available for each site. And then the background color shows the different climate zones that they fall into. So in total, we developed 33 new spring phenology models. And for that, we used the green update extracted from the PhenoCam data as our response variable. So the day of the year when spring green up began, we're trying to model that, to predict that. And the drivers we used for that were temperature, day length, precipitation, soil moisture. And we got most of those from large gridded networks that are available. So DayMet or NLDAS for the soil moisture. And then we put them in different configurations. So for example, we could require that a site needs enough moisture and then temperature to green up in the spring. Or a site might need enough warm temperatures in the spring and then moisture to green up. Or maybe it needs water and temperature, warm temperatures concurrently to green up in the spring. So that's just an example of some of the model configurations that we tried out. We used an R package called PhenoR, which was developed by a previous postdoc in our lab to fit all of these models. And so what this package does is it optimizes parameters using generalized simulated annealing and then determines the best model using AIC. And I'm just going to leave it at that. I'm not going to go super into depth into all the modeling stuff, but I'm happy to answer questions about it later. So we also fit, in addition to our new models, we fit some of these existing spring phenology models as a comparison. So for example, this is that growing degree day model that I discussed earlier, which uses temperature to predict the day of spring green up. So let me orient you. We have on the x-axis, the observed day of green up from the PhenoCam data. The y-axis is the day of green up predicted by the model. This is the one-to-one line, the dotted line, and the dark line is the regression line. And each point here is a site year of data, color-coded by what climate zone they fall into. So either arid, temperate, Mediterranean, or continental. And what we notice is that this model does not do very well. It has a root mean squared error of over a month of 35 days between observed and predicted green up dates. And it does especially poorly at predicting these early transition dates. So these are mostly Mediterranean grasslands, so California grasslands that green up early in the year. And these late greening up grasslands, which are mostly monsoonal southwestern grasslands. So we hoped that our new models did better. And they did. We found that all the new models that included water availability perform much better than any of the existing spring phenology models. And we found that the best model said that grasslands require both precipitation and warm temperatures to green up in the spring, but the order was important. So they must first receive sufficient precipitation and then sufficient warm temperatures for spring green up to occur. And there was a little caveat to that as well, in that spring green up was delayed if temperatures dropped too low. So think about having like a cold snap in the spring. If you had a cold snap, it delayed when spring green up occurred. So to review, a site must first receive enough precipitation and then enough warm temperatures for spring green up to start. All right, so let's see how that model did. This is our top model. And we see that it performed much better with a root mean squared error of 16 days between observed and predicted green up dates. It also caught those early and late transitioning grassland sites much better. And I want to point out that this is able to predict spring green up over 250-day spread in green up dates, which is pretty impressive. And this is all using a single parameter set. So one model for all of the grassland sites across North America. Unsurprisingly, if we fit that model separately for each of the 4 different climate zones, so we allowed the parameters to vary for each zone, we got an even better fit with about 10 days difference between observed and predicted green up dates. So the last thing we did is we ran our new top model using various future climate scenarios from Earth system models. And the goal here was to see how climate change might impact the timing of spring green up for grasslands from different regions of the US. So for example, all of these squiggles in the background, the gray squiggles, those are all of the results from feeding in the Earth system model data. So let me orient you. We have year here up to 2100. This is the day of year that spring onset is predicted to occur. And we have all of the predictions for each of these combinations of weather and temperature data that are predicted into the future. And then the black line is the average and the red line is the regression. So this, for example, is for a site in North Dakota. And using the slope of this regression line, we can see that spring green up is predicted to occur about 11 days earlier within the next 100 years at this site, based on that climate data. And we did the same thing for a representative site from each of the other 3 different climate regions. And we saw similar patterns. So this is a site in Kansas, and we see that it's predicted to green up about 5 days earlier within 100 years. And a site in California is predicted to green up 12 to 13 days earlier within 100 years. The only exception was an arid grassland site in New Mexico, which didn't show any clear pattern of advancement or delay in green up. And this is likely because this site is much more driven by water than by temperature. And predictions of future rainfall patterns are much more iffy. So maybe once we get better predictions of that, we'll be able to predict how these dry grassland sites will respond better, will change with climate change. All right, so to just review what we found from this project, we used PhenoCam Data to develop a new model that accurately predicts the start of spring across diverse North American grassland ecosystems. And it represents a substantial improvement over past grassland spring phenology models. We found that grasslands require both sufficient precipitation and warm temperatures, and in that order, to become active in the spring. And since plants are adapted to their local climates, we found that fitting the model separately for distinct climate regions improved model performance to within about 10 days of observed green updates. And we also found that our model predicts that spring green up will occur about 5 to 13 days earlier across most North American grasslands, and that arid grassland site was the only exception, which did not show a clear pattern. And so our hope is that we'll be able to integrate these new spring phenology models into larger Earth system models that could improve predictions of spring onset and all the associated ecosystem processes with it. All right, so before I conclude here, I just wanted to point out that we're happy to help you set up a camera if you want to add a new site to the PhenoCam network. It costs about $1,000 to get the camera and the setup for the site. Here's what it looks like. This is the camera here, and then this is the housing that it's in. Kind of looks like a security camera. And we have detailed instructions on the website. And what's cool is that once you set up the camera, you really don't have to do much to it. Your images will automatically be sent to the PhenoCam server, stored there, and then processed for greenness and available to see on the website. But I'm also happy to chat about it or put you in touch with other people on my team. This is Andrew. This is my postdoc advisor here. So happy to chat with you, or he can chat with you about any sites you want to set up. With that, I want to thank all of my past and present lab mates, my advisors, all the PhenoCam site collaborators that set up and maintain all of these sites. And I encourage you to check out the PhenoCam website or get in touch with me with any questions. Thank you.
Thanks, Allison. That was great.
Yeah.
Casey, are there any questions from folks in the room? We don't have anything in the chat yet. Oh, they're muted. I
think we're good.
Or, oh, sorry, go ahead.
Yeah.
Anything
Can you
from
hear
folks
that?
in the room?
Hello. Hey, Allison, this is Tyler. This is awesome. And I had a quick question about integrating this with these other models that you said have been used in the past. I know you said the growing degree day model tends to work well in more like northern latitudes and forests, those types of systems. Did you look at all at like under what circumstances this model works versus the other model, or did you really just look at the grasslands, the model right now?
So you wonder like different ecosystems, is that what you're asking?
Yeah, essentially like how and when we can start integrating these different phenological models across larger scales and across multiple different ecosystems, like the breakpoints at which different models begin to work versus others.
Yeah, good question. I have not worked with the deciduous forest models. That's actually, there's a huge sector of research that's already done that, which is why grasslands were kind of a low-hanging fruit to figure out a better model for. But I think there's going to have to be different models for different ecosystem types, just because of what you saw in that one graphic, that, you know, using just temperature, we can get a lot of the deciduous forest sites, but you need a little bit more nuance with water availability for these grassland sites. Did that answer your question, or?
Yeah, that's helpful.
Okay, sorry, I was trying to, I don't know if I quite understood it.
Okay, it looks like there's one in the chat here. Did you try to model the timing of senescence in the grasslands or just green up?
That is a great question. That's actually what I currently am working on. I have some good candidate models, and I'm actually hoping to be wrapping that up and writing it very soon. So stay tuned. But they also seem to be requiring some version of both temperature and moisture as well. I'll give you a sneak preview.
I have a comment, really, it's a comment, it's not a question, but I think it'd be really cool to come up with some learning modules that we could put on earthdatascience.org using PhenoCam data around like how, just some different ideas about using different measures of phenology in different ecosystems or something like that. I think that'd be really cool.
That would be awesome. Yeah, happy to help with that. And we already have some like, lessons worked out. So maybe we can like start to integrate some of those too. Yeah.
That would be super cool. Yeah. So maybe, we should talk to either Nate or Nathan and try to get them to help maybe update or come up with some new ones. That would be super cool.
That'd be awesome. Yeah.
Sweet. Yeah, anybody else in person or online? Questions for Allison?
I have a question. Yeah, thank you, Allison. I love your presentation. I have a question regarding the cameras. So do we have data on view angle, like the view angle, and also the direction of the camera, like the views? So do we have those informations available?
The metadata will always tell you the angle it's oriented, the direction it's oriented. We tend to recommend northern exposures just because you don't get as much shadow then. So most are north, some like to deviate from a protocol, but that will be noted. As far as angle of the camera, we don't have a clear protocol for that, just because it all depends on how high you can get above your canopy, right? So grasslands, you can easily get above the canopy, but for example, in those redwood forest sites, we actually mounted cameras to the trees themselves because you literally can't get above the trees without a huge tower. So the angle is not quite as consistent, but we haven't found too much of an issue, comparing sites, even if they're different angles between them. Good question.
Okay, thank you.
Yeah.
Okay, anybody else? All right, well, thank you so much, Allison. That was awesome to hear about the work you've been doing and are continuing to work on.
Thank you. Yeah, thanks for having me.
Yeah, thank you. Bye, everyone.